In the second section of Chapter 4, we're going to begin looking at the first type of primary tissues. So we're going to begin looking at the epithelial tissues. Now, to begin with, we have already kind of mentioned in the last section that epithelial tissues are sheets of closely adhering cells, typically one or more cells thick. They are found on every internal and every external body surface. So a good thing to remember about epithelial tissues is that they're linings. They either line internal cavities or they're lining external surfaces. Because they're typically found on surfaces, their main function is to act as a barrier between the body and the external environment. So you can find them lining organs, lining fluid-filled cavities, lining areas like your mouth and your nose, even lining your body found in your skin. In addition to being a barrier, the epithelial tissue serves several other functions, one being protection. So we just said as a barrier it's shielding underlying tissues from injury. It also carries out the function of immune defense. One of the things we'll talk about in anatomy too are ways that your skin prevents invasion. Some of the things that your skin actually secretes in order to protect your body in the form of, number 3 mentions hormones and oils that are secreted by glands that are found in epithelial tissues. We also know that epithelial tissues can function in support. A lot of them are selectively permeable membranes that allow some materials to pass in and out of them. And also your epithelial tissues are typically associated with a lot of nerves. So they detect changes in the environment externally to you. They also detect changes internally. For example, taste buds are a great example of a sensory structure that's found within epithelial tissue. So we looked at the functions of epithelial tissue, but now let's look at some of the components. So again, we mentioned consists of tightly packed cells. And typically these tightly packed cells are only tightly packed because they're held together by either tight junctions or desmosomes. So if you look to the picture on your right, you'll see at the top of this image, and this is on page 129, this would be epithelial tissue. So this is lining the surface, and you'll notice that these are connected by either tight junctions or desmosomes. And they're lining some type of internal connective tissue here. So because of their structural arrangement, typically they're impermeable or they're resistant to stress or to injury. Now, one key thing to remember about epithelial cells is that tissues that are epithelial are avascular. Now, we know if we put that word "a" in front of a word, it means without. So avascular means they're without blood vessels. Now the problem with that is we know blood vessels carry oxygen, carries nutrients, so if epithelial tissues don't have that, they have to depend on oxygen and nutrients to come from other areas of the body. And so what they typically depend on is a process called diffusion, and we mentioned this in biology. Diffusion just saying that solutes typically move from high to low concentrations until they balance out. So epithelial tissues depend on obtaining oxygen and nutrients by diffusion from tissues that are deeper to them. So if you look at this image, notice no blood vessels up here. So the tissues here would depend on getting oxygen and nutrients from the blood vessels in deeper connective tissues to them. Now underneath underneath each epithelial tissue, you'll find what's called a basement membrane. And so easy way to remember it is to think about just a house in general is going to have if it has a basement, the basement is underneath it. So the basement membrane typically found underneath it. And it's made of two main things, what's known as the basal lamina and the reticular lamina. Now both of these together kind of glue or anchor the epithelial tissue to what it's lining. So if you look at the picture again, you'll see this is the epithelial tissue, here's the underlying connective tissue, and there's the two layers that form the basement membrane that anchors this on. So a very important feature that we find. Another thing we want to look at with epithelial tissues is how we classify them. Typically they're classified based on the number of cell layers as well as the shape of the cells. So as far as the number of layers, we use the terms simple and stratified to define how many layers are present. So if we say that an epithelial tissue is simple, then we're saying it consists of a single layer of cells. Now typically these single layer cells are so thin they're very good at transporting substances through them. If a epithelial tissue has more than one layer of cells, we typically call it stratified. So simple epithelial for a single layer of cells, stratified for more than one layer. And then based on the shape of the cells, we use the terms squamous, cuboidal, and columnar. So squamous are cells that have been flattened or kind of squashed, if you will. Cuboidal cells are short, typically square or round shaped. And then columnar cells or columnar cells are typically tall and skinny, like columns. So if you look on 129, you'll see here's one layer of cells versus multiple layers, so simple versus stratified. Here's the squashed squamous cell, there's the round or square cuboidal cell, and then there's the tall and skinny columnar cell. Now, we just mentioned that the simple epithelial, they typically are so thin, they're really good at transport. So two major types of transport that we see in epithelial tissues are either paracellular transportation or transcellular. So paracellular is where substances leak between cells. That prefix para, we'll learn this semester, means beside. So this paracellular transportation is where materials move beside cells, so in between one cell and the other. The problem with this is it's limited to tight junctions that may not permit movement of material. So you'll see here paracellular typically goes beside the cells, so in between them. Transcellular, that trans means to go through. So transcellular transportation is where substances actually go through the cell. They diffuse in one side and out of the other side of that cell. Now, one thing to keep in mind when you're looking at these pictures is we're about to look at the different types of epithelial tissues. And one of the things you're going to notice is that they all are colored pink. So the question is, well, how do you distinguish one from the other? And so the main things you're going to look at is you're going to look for the types of cells, and you're going to look for the extracellular stuff. So the key thing is anywhere you see a dark purple nucleus, that's a cell. So you see a dark purple circle, that's the nucleus, meaning that's found inside of a cell. Ground substance typically either looks clear or lightly colored, and then protein fibers typically are going to look like wavy or straight lines. So keeping all that in mind, let's look briefly at the different types of epithelial tissue, and that'll get us through this section. So the first type of tissue is called a simple squamous epithelium. So just by the name, simple squamous, you know it's going to be one layer of very thin, flat cells. Typically we say it kind of has a fried egg appearance. So if you look here, this is that one layer of fried eggs, or one layer of squamous cells. Underneath here would be all the underlying connective tissue. Again, it's very, very thin, so this type of tissue is adapted for rapid diffusion, which is why typically it's found in areas where materials need to move in and out quickly, so the lungs, the kidneys, as well as blood vessels. So again, keeping that fried egg in mind, this would be the simple squamous. The second type of epithelium we see is simple cuboidal. So again, by the name, simple, it's going to be one layer, cuboidal, cube or round shaped, typically having a nucleus that's very large and in the center of it. So you see here's our one layer, and then there's the basement membrane. All this would be connective stuff. This type of tissue is still very thin, so good at rapid diffusion, good at absorption and secretion, typically found in areas where you're going to be absorbing or secreting materials. So renal tubules will be in the kidneys, respiratory passages and ducts, also associated with some of the major glands of your body. So simple cuboidal would just be one of these layers of cube-shaped cells. Simple columnar, again by the name, one layer of tall and skinny cells, so rectangular shaped. Typically the nuclei is located towards the basal portion, so towards the bottom here. Couple of things that make this one different is you often find microvilli on the apical surface, so towards the outer part here. Typically you'll find some microvilli here. And so this type of tissue typically found in like the small intestine as well as the respiratory tract, where microvilli or even ciliated cells are used in order to move substances through. An additional type of epithelial tissue we see here is pseudostratified columnar. Now, that prefix pseudo means false or fake. So this particular tissue looks like it's layered because the cells that make it up are different structures and shapes, but actually every single cell touches the basement membrane, so it's only one cell layer thick. So this is that kind of fake one that looks like it's multi-layered, but it's actually not. This tissue does possess cilia, you can see those hairs here, and it possesses goblet cells that are used to secrete mucus. So typically found in areas like the respiratory tract, the nasal cavity, also can be found in the GI tract in moving materials. As we continue looking through the epithelial tissues, we come to the stratified squamous epithelium. Now, stratified squamous meaning many layers of flat cells, but there's actually two types of stratified squamous. The first is keratinized stratified squamous. Keratinized, typically the apical cell layers, or the further away you go from that basement membrane, the more dead cells you're going to find. They're lacking nuclei, they're lacking nutrients, which is why they're dead, because they're getting pushed further and further away. Remember, all this is avascular. Keratinized means they are filled with the protein keratin, which makes them tough, it hardens them, and they're well adapted to act as a barrier for protection. So typically this is going to be found in the outer layers of your skin. The other type of stratified squamous is non-keratinized. So non-keratinized, we typically see some of the ones closer towards the bottom are going to have the nucleus, they're going to be alive, but the big thing here is they don't possess keratin, so they've not been hardened. Even though they are still very strong, they remain moist. So this is found in areas like the mouth, the throat, the esophagus, the anus, the vagina, areas where they need to be tough materials but are going to remain moist because they've not been hardened by keratin. We also find stratified cuboidal, even though it is rare in humans. It consists of two or more layers of the cube or round shape, and typically found in your gland region, so sweat glands, the ovaries, as well as the testes and the seminiferous tubules inside of them. And then the stratified columnar, which also is rare, but again looks like multiple layers of columns or elongated cells, typically found in the male urethra, the cornea of the eye, and even the salivary glands. Last epithelial tissue we want to look at briefly is the transitional epithelium. Now, to transition means to move or to change. So this type of tissue only found in the urinary system, and it lines those organs that need to change shape. You'll notice that the basal layers are cuboidal, where the apical are dome-shaped. And so when the tissues relax, it takes on that, meaning the apical cells can kind of flatten and then allows that tissue to stretch. So your bladder has that ability to kind of stretch in response to filling with urine, and then to recoil once that urine is expelled. One other component we want to look at with epithelial is we've mentioned glands several times as far as locations of these tissues. So just reminding you that a gland is any structure of epithelial origin that's going to synthesize and secrete some type of product, specifically some type of solution. And so the two main types of glands we see in the human body are endocrine glands or exocrine. And just looking at brief differences here, the endocrine glands, endo for inside, so these secrete their hormones mainly, or secrete their products, mainly hormones, directly into the bloodstream. So they don't use a tubing or duct system straight into the bloodstream. The exocrine systems do use a tube or duct system. So they typically are going to release products onto the surfaces of epithelium that at some point are going to open to the outside of your body. And again, they do so using a duct or tube system. We've also mentioned goblet cells as being found in some of the types of epithelial tissue, and again we've mentioned their job is to secrete mucus, so protecting underlying epithelium, also aiding in the movement of materials through these different organs and organ systems. One of the last things we want to look at with glands is that exocrine glands have two methods for secreting products. The first method is the merocrine secretion, which is what a majority of the exocrine glands are going to do. But they package their products in vesicles, and then the vesicles are released. So you'll see that the products are put into these small vehicles, if you will, and then they're released through exocytosis, where the vesicle opens up and releases the material. The second way that they release their secretions is a holocrine secretion, which literally says eventually the entire cell ruptures and is released. A good example of this would be sebum, or the oil that's produced by sebaceous glands.